All right, so welcome to our video. Oh, what you present? There we go, on enzymes. So enzymes, we learned when we were talking about protein functions, are a type of protein molecule. What we're going to do now is have a little bit of a mini unit where we're going to discuss enzymes and their role in the human body, or all living things actually, a little more in depth. And we're going to combine that with understanding how the digestive system works, because the two are very closely related. So our question is, how do enzymes increase reaction rates? Now, the overview, we're going to watch a short video. It's about 4 minutes. Answer a few review questions, and then once you're done with these, you can start your homework. You should not be working on your homework while you're doing the video. Do one at a time. They do build on one another. So here's your video. It is goofy, so I will warn you that. However, the information in it is very good, so please watch it. Enjoy.
Enzymes.
Hey, Professor. Just the other day, I saw this small man swallow an entire steak bigger than the size of his head. And it got me to thinking, how could he possibly digest all that in just a few hours?
Well, the steak is first chewed in the mouth into bits and pieces that fall into the stomach. You might already know that the stomach contains a powerful acid that can dissolve even metals. But what you did not know is that this acid is not what ultimately breaks down the steak. No, it's very tiny and special molecules called enzymes that do the job. Strangely, these enzymes find the stomach acid a cozy place to work in.
Whoa! It's like small piranhas finishing up a whale in a sea of acid. Imagine what I could do with that!
Ooh. Huh?
Well, it doesn't work that way.
What do you mean it doesn't work that way?
I knew you were going to say that. To find out, you need to know what exactly enzymes are and how they work. Whenever you're ready, click Next to continue. What are enzymes? They are proteins that speed up chemical reactions. Without them, reactions can still occur, but they would be too slow to support life. There are many different types of enzymes, and they come in all manner of shapes and sizes. For example, what you call the piranha enzyme is actually pepsin. It is only found in the stomach and digests proteins. In your mouth, there is amylase, which breaks down starch. In your cells alone are hundreds of enzymes that control your cell activity and defend them from invasions by microbes like bacteria and viruses. Though enzymes can appear very different, they all share a few characteristics in common. Let's now take a look at what these are. Number 1. Each enzyme has an active site. This is the groove in the enzyme where the molecules they act on, called substrates, are captured and made to undergo a reaction. The substrates are either broken apart or combined together to form products. For instance, starch found in the bread that you eat is digested to form simple sugars called glucose that is then absorbed into the body. Glucose is used up as energy, but when there's too much of it, special enzymes combine the glucose molecules together to form a giant molecule called glycogen, which is then stored away in the liver and muscles for later use. Number 2, enzymes are very specific. Each enzyme can only bind to one type of substrate. Let's look at the case of lipase enzyme, which breaks down fats. Molecules like glucose and proteins cannot fit into the active site of lipase. Only fat cells can bind to the active site because they have the right shape. This ability to act on just fat cells is very important for lipase. Lipase converts fats into energy. Now imagine what would happen if lipase can also convert proteins into neurochemicals for the brain to function well. If you no longer need the extra energy from your storage of fats, lipase will be deactivated. But this means that lipase can no longer produce precious neurochemicals for your brain. In other words, when you stop burning fats, you end up damaging your brain. To prevent such freakish scenarios from happening in our body, enzymes must only act on one type of molecule. Number 3, enzymes are recycled. Enzymes are not altered by the reactions they speed up. Immediately after they finish one reaction, they can act on a new substrate. This way, only a small amount of enzyme is needed to speed up thousands of reactions.
All right, so hopefully you guys liked that. So now let's check in, see how well we understand the information that was in this. So question number 1, how would the presence of enzymes impact a chemical reaction? Good, so hopefully you guys were able to get that enzymes are going to speed up the rate of chemical reactions. Now these reactions would occur on their own. However, in order to keep organisms such as ourselves alive, you need these reactions to occur at a very sort of quick pace or a higher rate than would occur naturally if we were just to let things wait around until they bumped into each other in order for a reaction to take place. So enzymes are going to kind of speed that process up. Enzymes are also called biological catalysts. So if you see this word catalyst around, catalyst is just a molecule that is going to speed up a chemical reaction. So you might see this term or come across it in your reading as well. Question number 2. So right here we have an enzyme. That's this green structure right here. So the first question I want to ask you is on your enzyme, what do we call this place right here where something is going to connect to an enzyme? Good, so hopefully you were able to get that this is called the active site. The active site is the location on an enzyme where it's able to receive a molecule. Now, next question, this purple thing right here, what do we call these molecules that are going to bond with our enzymes? Good, so hopefully you were able to get that these are substrates. So substrates are molecules that will bond to the active sites of our enzymes. Enzymes are going to help chemical reactions along by either splitting these substrates apart into components, or they may combine substrates together to form a product or a final molecule. So here is an image of your enzyme substrate complex. This is a term that just describes when you have your enzyme and your substrate with the substrate bonded to the active site. And the common analogy that's used to describe this structure, because the substrate of a specific shape will sort of bond to the specific active site on your enzyme, that's referred to as the lock and key model, since they're structurally dependent on one another in order for this complex to form. So enzyme naming is just a little practice, not something I would kind of expect you guys to be able to do too much of. So A, what does the enzyme lipase break down? So try and see if you can remember this from proteins. Good, so lipase is going to break down lipids. B, what does the enzyme sucrase break down? Good, so hopefully you're getting the hang of this. All right, so sucrase is going to break down sucrose. And C, what does the enzyme peptidase break down? This one's a little trickier. Good, so peptidase is going to break those peptide bonds in your polypeptide, so it's going to break down your proteins into amino acids. So the reason I did this is because I wanted you guys to notice what suffix or ending of the word do you notice is found in all of your enzymes, lipase, sucrase, peptidase. Good, so hopefully that was an easy one. It's the ACE ending. So ASE on the suffix or ending of a word is a naming convention that lets you know that that molecule is an enzyme. So it's a biological catalyst, a molecule that's going to speed up a chemical reaction. The root of the word is always dedicated to whatever it is that enzyme is breaking down or building up. So in that way, we use that naming convention to help identify the functions of our enzymes and sort of identify them in readings much more easily. So what you guys are going to do now is you are going to work on your homework, which will sort of take these basic ideas and it'll be a little more in-depth. Once we're finished with your lab, we'll come back and spend a little bit more time on enzymes, but finish this both for next class.